the chat up. Okay, so this is just my generic starting slide because I'm here in the Digital Microfluidics Laboratory. Right, so the purpose, I think, of today's talk, there's sort of two main purposes. Right here, I'll turn my video off here. So two main purposes, right? The first is to give a bit of an overview of the work that I've done, right? And I think that the reason that I want to do that, the reason that we're here, is we're trying to sort of open the door to new collaborations, right? So we want to be able to help each other. So I think for some of this, it's worth having some context of kind of the path that I took anyway, right? So I started off, I did my undergraduate and my master's degree at McMaster University, which is in Hamilton, Ontario. The topic of my master's degree was looking at capillary evaporators. So in capillary pumped loops. So here we have a porous wick, which would be filled with fluid, and we have heated fins that come down towards the wick, and then boiling happens at the fins and then comes out these vapor channels. So here I had two papers. The first one looked at what happens when we add a gap between the wick and the fin, right? So obviously Satish knows more about this than I do, but basically when you add a gap, it's easier for the vapor to escape, so you can push more heat into the evaporator. Then we also looked a little bit at pore size inside the wick here. So after that, I went to the University of Toronto, where I started my PhD and then did my postdoc there as well. And what I was interested in there were these digital microfluidic devices. So basically, we use electric fields to move droplets around on these checkerboards, right? So we have this checkerboard of an electrode, no channels, pumps, or valves, but we can move droplets around. So for a sake of size, I don't have a scale bar here, but these electrodes can serve as your scale bar, and they're typically on the order of 1 millimeter by 1 millimeter. And then what we do is we sandwich the fluid between these two plates, right? So the droplet looks like the white part in an Oreo cookie, right? So both of these plates are hydrophobic. The gap distance here is on the order of 100 micrometers. So that's what makes this a microfluidic flow. On the bottom substrate, we have this sort of checkerboard of electrodes where we can kind of control where the different where the electric field goes. And then the top plate is something that's transparent, so maybe glass, or we've used polymers, and it's coated with indium tin oxide, which is a transparent conductor. So we use this as a large ground electrode, we cover it in some Teflon, and then we can do things like move these droplets around. So in my PhD, this was mostly fundamental stuff that we did. So we looked at sort of what drives these droplets, why do these droplets move when you apply an electric field. The nice thing about the geometries here is essentially when we have a droplet in a position, it's essentially a parallel plate capacitor. So you can measure capacitance or the inductance of the droplet, which is a combination of the capacitance and the resistance. And you can measure things like we were looking at fluid composition, so to measure droplet mixing. And then later we measured some chemical reactions and particle concentrations inside these droplets. We also developed a way of mechanically filtering droplets, so doing separations between the fluid in the droplet and particles in a droplet, basically with a comb filter. So if people want to talk about that, we can talk about that. Also in my postdoc, I had the opportunity to work with a company called SQI Diagnostics. They were interested in microarrays to do things like immunoassays. So they were basically looking for the presence of proteins, so diagnostic testing, right? And one of the problems that they had is they have these little droplets, but when they dry out, they get this sort of coffee ring shape. So we started to look at this in my postdoc, published a paper in Langmuir. And then sort of throughout my PhD and my postdoc, I also, through a network that I sort of developed myself, a friend of mine from undergraduate was working for a company called Connectrix. So they used to be part of Ontario Power Generation, but then they got split off and now they do consulting for Ontario Power Generation, right? So I did some CFD stuff with them. So this isn't sort of my core competence, but I know a little bit about how to do CFD simulations. Then I came to the lovely town of Rochester, right? And I founded the Discrete Microfluidics Lab, right? So I think that this goes to a little bit what Satish was talking about last week, where I think the way that I try to approach the work that I'm doing, and maybe the way that we all do as engineers, is, we're trying to have a better understanding of the fundamental aspects of these problems for the purpose of then making impact in important societal problems, right? So we're interested in things like lab on a chip devices. So my brother's a biochemist, and I always kind of joke that I'm trying to put him out of business, right? So he's one of these people in coats that's walking around doing lab stuff, right? And we're trying to shrink that down onto a microchip so they can be more interested in the science and less interested in sort of the day-to-day, pipetting stuff into reservoirs. I have a story about that too if people want to talk about it. I worked in an intestinal disease research lab in high school. I won this competition, but if you want to know, I'll tell you the reasons why I picked mechanical engineering over biology. We're also sort of continuing some of the work that I did with these droplet evaporation experiments. We think that this has some utility in microarrays, but also potentially in trying to reduce feature size in inkjet printed electronic devices, so for flexible electronics. I did some work with Patricia looking at energy harvesting. So I know Satish was talking about the potential of trying to put some things together for building efficiencies. So maybe there's an energy harvesting component in there. Most of what I do, you put an electric field in and you get mechanical motion. But at least theoretically, you can take mechanical motion and turn that back into electrical energy. So we sort of looked at that. And then this has been mostly led by Patricia, but I've been sort of helping a little bit looking at lubrication and the role of wettability in lubrication, particularly with ionic liquids. So at the core of what I do is electrowetting on dielectric, right? So, or EWOD, as some people say, right? So here we have a droplet, right? Our droplet is sitting on a hydrophobic surface. So this contact angle is fairly large. Often we'll deal with something like Teflon, right? So maybe the contact angle approaches 120 degrees. Now, so underneath my droplet, I have this sort of hydrophobic layer, but also some kind of dielectric layer here, and then an electrode. I need to ground the droplet somehow, and the simplest way to do that is to take a really small wire and stick it into the droplet. There's other ways to do this, but at least, conceptually, this is probably the first way that it makes sense to think about. Then what we do is we apply an electric field. Now, after we apply that electric field, what happens is we apply a force on this droplet interface right above the contact line. So what happens is if you think about, the skin of the droplet, almost like a balloon skin, we're sort of pulling out the skin of that balloon so we change the shape of the droplet. And that's related to the capacitance per unit area and the voltage that we're applying. So the RMS voltage that we're applying here. So this is pretty well predicted, at least until you hit some saturation limit. So we can change the shape of the droplet. What's neat about this particular problem is that, if you're a mechanical engineer and you look at this, you might think that, oh, well, the droplet, that skin, that balloon skin is a bit like a spring, right? There's some elasticity to it. So if I understand the dynamics of how the droplet moves and I apply the right kind of electrical pulse, what happens is, if the droplet's underdamped, it overshoots its equilibrium position, and there's some extra energy stored in the interface, like a spring, and it'll bounce back up off the surface, right? So this is we took this at 600 frames per second. It looks like almost like a droplet of Jello or something bouncing around there. It's not quite as nice. Satish always has those really beautiful pictures of droplets that he takes with the beautiful imaging system that he has. So I don't think it's quite as cool as that, but I like this video, so you'll see it a lot in this presentation, right? Now, one of the downsides of these types of devices is that, at least on a laboratory scale, they get to be reasonably expensive for us to make. You got to go, throw one of those ninja suits on, go into the clean room, and anytime you do that, the expense goes up. So we also have been working with Kathy's lab, right? And we sort of applied for some internal money and got this kind of inkjet printed system. So this is just a printer we bought off Amazon. We load it with some conductive ink, and then in Kathy's lab now we have a spin coater. So, we spin this, the polymers that we want on here so it's hydrophobic, and we can do some of this device fabrication at a lower cost. Right, so I think what's cool about electrowetting on dielectric, so it's this microfluidic platform that we can use to move lots of different types of fluid, right? Particularly biological fluids we're sometimes interested in because we're interested in this kind of lab on a chip space. And it's neat because, sometimes when you're doing microfluidics, you kind of have this very small device, but then lots of equipment around it. So here we don't have things like pumps or valves. It's just this sort of two-dimensional plane that we move the droplet around on. Like I said, the system geometry, because we basically have a parallel plate capacitor for every addressable position, so that enables some real-time feedback without having to look what's going on in the device, even though in this embodiment we can see the droplets. And there's also been, so one of the things in microfluidics, particularly in digital microfluidics for a long time, there was always this argument that, oh, it's kind of cool, right? It makes neat videos, but does anybody really care about it, right? So there was a company, Advanced Liquid Logic, which was spun out of a research lab at Duke, and it was purchased by Illumina. Now, Genmark is a subsidiary of Illumina, and in 2019, they reported $88 million of revenue using these digital microfluidic devices. So there's hope that there's some commercial relevance of this particular technology. Another way, so, I've talked about kind of squishing these droplets like Oreo cookies, right, and moving them around. But there's another way that people have done this where the spacing between the two plates is a little bit further apart. So here you can create droplets, move them around, and then jump them up to this other surface, right? Remember I said, if we understand the dynamics of the actuation, then you can sort of get to this point where you can sort of have these droplets jump up, right? So here's a side view image that was taken. This is not our work, but this video is our work. And I think it was Michael Schertzer who noticed last week that we tend to get good news when we have these meetings. So it was technically last night, but I didn't check my email till this morning. But we just had a paper where we develop or improve upon a model for this droplet jumping, and it was accepted at Langmuir late last night. So I'm going to count that as today because I only saw the email today. We're also interested in contact line motion. So this ultimately for when we're evaporating droplets, this becomes important. We really like this paper, which was published in 2008, in part because it's got a great title, How to Make Sticky Surfaces Slippery. And they were talking about kind of the elimination of contact angle hysteresis using AC electrowetting. So we looked at their model and saw that it didn't quite predict the data correctly. So we proposed a change to the model using the data that was published in this paper. So this was a comment we published last year in Applied Physics Letters. And then as we were doing that, so we did some analysis using their data and then started to do our own experiments, first on Teflon surfaces, which is kind of repeating basically what was in that other paper. You can see the dotted red line is the old model, the model that's in that other paper, very well-cited paper, excellent paper. And this dashed black line is our model. So you can see we predict, this is the cosine of the advancing contact angle. We predict this much better. This was on Teflon, but we wanted to show that it works on all kinds of different surfaces. So we also get much better predictive value on different surfaces like SU8, which is a photoresist, and on PDMS. So this work, this was done by Kim Bernetsky. So I don't know if people remember Kim. She graduated a little while ago. So, I've been left with this data trying to put this paper together. So I think we have a version of this paper that's just about ready to submit. So I'm hoping to be able to submit this back to Applied Physics Letters by the end of the summer. So we also talked about low-cost fabrication. So here we wanted to, we took these, so we read a paper about this too, and we tried to improve the process. So we start with this inkjet printed device, or inkjet printer. You can get this on Amazon for maybe, I don't know, just over $100. We load it with some ink that's conductive. So we have to take out the ink cartridges that come with it and then replace them, right? And then we can print out, right? You just make a file, you print out different electrodes. This is a big electrode here, but I'll show some work that's on printed electrodes where, we're making electrodes and moving droplets around. Then we spin coat this. So we do this now in Kathy's lab. So this is outside the clean room, which is nice. So it's less training for the, because the clean room, you do have to go through some rigorous safety training in there, and it's definitely warranted for the work that gets done in that lab. But the stuff that we do is, they still go through safety training, but they don't have to do the whole clean room safety training to get trained for things that they're never going to use. So this is all nice because you never have to step foot in the clean room, right? So this is a lower cost way to do this. We published a paper about this fabrication method in microfluidics and nanofluidics. Right? So getting back to like the fundamental stuff is great, right? As an engineer, this is the stuff that I really like. I really like trying to understand the physics a little bit better. But we do this because we think it can make a difference, right? So I think most of the work that I do is kind of in this lab on a chip space or in this droplet evaporation space, right? Oh, this is Evelyn. Just a sec. Daddy's gonna finish his talk, okay? So here, right, the reason that we like these digital microfluidic devices, right? They're digital because we're moving around small packets of fluid, right? So the idea here is that we can automate complex biological protocols. So, if you have lots of different reagents, this type of a solution is nice because you don't have to have lots of different channels, right? So you can deal with lots of different reagents and run them over this 2D array. You can multiplex things a little bit easier in these kind of devices. And when we say reconfigurable, that means different droplets can travel over the same path, right? So we don't need separate channels for different reagents. So we can get high throughput, low reagent consumption, and low cycle times. We can simultaneously manipulate lots of different droplets. So this is like, I always tell people, and even though this is like the oldest reference, so, I was never around when they did this, right? But if you think about those old telephone operators, right, with the big jacks and they're plugging them in one after another, right? So we basically have a matrix switching capability like that where we're sending different electrical signals to these different electrodes to move droplets around. And we can do that we can move lots of different droplets at the same time. There's no mechanical components in here. So channels, pumps, and valves, you don't need those for this type of a device. And we can get real-time impedometric sensing of things like chemical reactions as they're happening on the chip. People have also shown that you can, this type of actuation scheme is compatible with lots of different biological reagent devices. So you may remember this slide, you may not too, from my tenure presentation, right? And this was an inkjet printed device where what we were doing, we were taking two droplets, right? Now I'm a mechanical engineer, so I think about this DNA, building stuff like Lego. So we have kind of one brick of DNA in the blue droplet, one brick in the red droplet. We create droplets, we bring them together, we mix them together, and then we get, hopefully the chemical reaction takes place and these two like DNA bricks, which look like Lego blocks, come together to make a longer DNA brick, right? So this was something we did on an inkjet printed device. We brought these droplets together, we mixed them around. We did gel electrophoresis, right? So here we had two different bricks, right? So these are two small double-stranded pieces of DNA. We mixed them together and we got one large DNA piece, right? So this was good, right? And at the time of my tenure package, I said, well, it's nice that we showed that this worked, but what we want to do next is try to scale this up, right? Because, the inkjet printed devices are nice for proof of concept, and, moving stuff around, two droplets at a time, it's good. It shows that it works, but we want to be able to scale things up. So this is, plug and play. This is maybe like 8 inches by 4 inches, and it plugs into your computer like a USB, and then you can control devices like this. So this is taken with this webcam, right? And now here, right, we're taking a droplet of the blue and a droplet of the red, which contain different DNA pieces. We mix them together on the device. You can see that the mixing scheme we chose here was not necessarily great because the droplet keeps splitting in this case, right? So we now picked a better actuation scheme, right? But here, this can control up to 120 different electrodes, right? So we've done this DNA ligation with droplets where the gap was filled with air. So that's in this particular figure. So these we're creating and mixing droplets in air. And this is controlled with not this dropbot system, but a National Instruments PXI box that we have. So that's more scalable if we need more than 120 electrodes. And then also, so this dropbot system is made to move droplets around in air. So we had to kind of modify our production of things in the clean room to be able to seal this device up fill it all with oil, so like a silicone oil, and then we're, loading droplets and moving them around. So the reason for doing that is we reduce biofouling so that we don't get, deposits of this biological material on the surfaces. And then if you have a process that takes a little longer, you also don't get evaporation. So because we needed to do incubation on this device as well, so after we mix these droplets together, we have to incubate at 37 degrees. So here we have a foil heater, which I believe that my students got from Satish's lab. We calibrated the applied voltage to the temperature of a droplet. So we basically had a reservoir filled with oil, deposited a droplet here underneath with the heater underneath, applied different voltages, and got different steady state temperatures. So we don't need open loop control for this temperature control. We can just pick a voltage and that'll give us a temperature. And then here, this shows that in both air and in oil, we were able to take these two bricks, mix them together, and we get the same size piece of ligated DNA that we did when we did it on the benchtop. So this is nice because it shows that it works, right? So now what we're trying to do is scale this up even more. So first we want to be able to make DNA bricks out of oligonucleotides, and then we want to stick those bricks together to make very large DNA products. We also have a couple other projects in this lab on a chip space. So the first one came out of this idea that we have in Lab Talks, right? So Mike Schertzer's, I'll say, had the pleasure of sitting through presentations from my lab a lot. And I've also had the pleasure of sitting through lots of presentations from his lab, right? So, I know about moving these droplets around. Michael knows about impaling cells on these nanotubes, right? So we said, why not put them together and make kind of a device that, takes advantage of both of the advantages of these different technologies. We're not going to talk too much about the nuts and bolts of how this works. We have an invention disclosure that we're just about to submit. So we're hoping to get some IP out of that project. I also, right, so, we talked about new collaborations. So Kay and I sat together maybe 2 weeks ago or so, and we're thinking about how can we make a coronavirus detection device. So a diagnostic device for coronavirus, something that can be used at the point of care, right? So again, like, I like to move droplets around with electric fields. So this, we sat together and we talked about this. And so the idea here is a multiplex thing where you could take many different samples and test them for coronavirus, right? So what we do is we take in sample one, so this is a sample, maybe it has RNA from the coronavirus, and then we mix it with this RPA reagent, right? So RPA, maybe you've heard of PCR, which is something that we use to amplify DNA. So RPA is a similar protocol, but it happens all at one temperature instead of having the thermal cycling that you see in PCR. So that makes the device easier if you can do it that way. So basically we take this RNA, we make some complementary DNA out of it, and then we replicate that. And then we get to sort of Kay's core competency, or one of Kay's core competencies, right, is then how do we detect whether or not this thing that we've created is the result of this viral RNA in the sample. So was there viral RNA in the sample? And so Kay has some experience making CRISPR assays to detect whether or not certain viral pathogens are present inside of a sample. So this is, we just submitted this on the 16th. So it's been a busy week and a half or so for us, but things are in now. So it always feels nice when you hit that submit button. So we're also, I don't know how many people saw the email, but we're also thinking a lot about Noah, who I guess had a house fire. So he's dealing with some significant issues. Another area that I'm interested in is this idea of colloidal transport for evaporating droplets. So here we have a droplet. It's got some stuff in it, right? So where do the particles go when things evaporate? So maybe you have this radial flow that gives you that coffee ring pattern. You can have a Marangoni flow, which tends to mix things up. If the interface collapses very quickly, so if you have a droplet evaporation that happens fast, particles can get stuck on the interface. So you get this interface capture and something that looks uniform. And then you can have these DLVO effects where basically if you have a charged particle that's an opposite charge of a surface, then they'll attract. And if it's the same charge, then they'll repel, right? So lots of people have looked at different mechanisms in this type of a problem. But not surprisingly, maybe I'm interested in what happens when we apply an electric field, right? So here, there's a couple things, right? So first, we get this dramatic change in the shape. So the interface almost instantaneously drops significantly. So we think interface capture might be important here. We get a change in the contact line dynamics, which we talked about a little bit already. And if you get your frequency high enough, you can have electrothermal mixing here. So basically, you heat up the fluid near the contact line here, and that causes some mixing inside the droplet. So there's lots of different things that can happen in here. There was a paper published in 2013 that said that they had some electrophoretic mixing with a manipulation of particles with a DC field. Basically, what they said was, if they have charged particles and they apply a DC field, they can suck the particles down. I thought about this for a while, and we were trying to do this, but we could never repeat their experiments. And I think it's because this doesn't actually happen. So if you look at simulations of the electric field, if you apply a DC field, the field never penetrates inside the droplet. So I don't think you get any electrophoretic manipulation in here. They said they saw more uniform deposition patterns here, and I think that's because they got significant interface capture when they actuated the droplet. So the interface came down instantaneously, trapped a bunch of particles so that they couldn't get to the contact line, so they didn't get this kind of ring-like deposit like you'd see if you cut open a tree. We're also interested in what happens if we apply non-uniform electric fields. There's been some papers like this one from Zhao et al. In 2007. I drew the figure myself, though, right? Where they were basically collecting particles on one side of the droplet using dielectrophoresis. So we think we might be able to do a similar thing to control where particles go inside the droplet. We've made some contributions here, too. So some of this is repeated, right? So that the comment that we made on the paper in APL, the characterization of contact angles and things like hysteresis on inkjet printed devices. And then Colin, so this was a special edition of a paper from a conference that Satish had in Norway. So we published a paper here that was looking basically at this interface trapping that happens when droplets are evaporating. We have this idea where if we could better understand the physics of what's going on here, particularly when we apply electric fields, maybe we could detect the presence of certain pathogens. So we're working with Leah Michelle on an early sepsis detection device where we have sort of this immunoagglomeration of particles. So if the marker that the protein marker for sepsis is in there, then the particles clump together, and basically you get a different kind of deposition pattern than if the particles didn't clump together. Just really quickly, the work we did in energy harvesting. So like I said, usually I apply an electric field to a droplet and get mechanical motion. It's possible to turn mechanical vibration back into electrical energy. So I wrote a model for this, so an analytical model. We didn't have the data to sort of validate it, but we sort of have an idea of how this works. A couple years ago, Patricia, myself, and Tom Smith worked on this. Instead of saying droplets, Tom knows about hydrogel beads. So we made different kind of hydrogel beads and played around with the properties using ionic liquids and then some metallization processes. So we have the model for how one of these devices might work, and then some interesting ways to make beads that might work as proof masses inside these types of devices. So again, this is Patricia's lead on this, but we looked also a little bit wettability and lubrication. So I certainly am a novice in this area, but I know a little bit about wettability, so I've tried to contribute to a couple works in this space as well. So we talk about trying to make collaborations. So I just want to highlight some of the collaborations that I already made. So I've submitted proposals with Dr. Charlot, with Blanca, with Leah Michelle, with Kay. So with collaborators at URMC and at Nuclera. In the colloidal deposition, so a lot of this work is done with Karamaki, but also for that proposal we were talking about with the different deposition patterns, we talked to Blanca and Leah there. I've worked also with SQI Diagnostics and my former supervisors at the University of Toronto. In the energy harvesting and lubrication stuff, so this is, again, working with Patricia and Tom on both of these projects. And then through Patricia on this lubrication stuff, I've also had the opportunity to write some papers with some collaborators in Spain. So this is my last slide, and this is part of the reason that I'm recording it because I kind of wanted to talk to Risa about this, and maybe we'll have the opportunity to do that in the future. But I went to a conference in Hawaii, not last summer, but the summer before, and I was listening to a presentation from a professor there who also graduated from the University of Toronto. And he was talking about this method of measuring surface tension that they sort of developed in their lab, and they were selling different components to do this surface tension measurement. And what was really neat was they had this enclosure, and they could add different pollutants inside their enclosure to see what happened to surfactants in, say, lungs, right? So in Hawaii, they're interested in volcanic ash and what that does to breathing. So in this device, you could do something like that, right? So here there's confined droplet measurement, and they're using that to measure surface tension and how it changes when these surfactants are exposed to different pollutants. So I've been thinking since we started to look at sort of the dynamics of this droplet actuation when you just sort of hit it with a pulse of a DC voltage, I think the dynamics here are a function of the surface tension, the viscosity of the fluid, the volume of the droplet, and probably some other things. But it might be possible to make a pretty low-cost device where you just have a stage, a droplet, a backlight, and a camera. So the camera we have is a couple $100, and it can shoot at 600 frames per second. So we can measure the overshoot and the time that it takes to get to that overshoot reasonably well. So if we could do something like that, maybe we could have a pretty low-cost device to measure surface tension in an enclosure where we can sort of have similar pollutants. So maybe with Risa, we can answer questions like how does vaping affect the surface tension of lung surfactants? Maybe if it's sort of COVID specific, how does one sec, how does having the virus and the disease associated with the virus, how does that affect the surfactant of these lung surfactants? And is there a difference between the effect on these lung surfactants between smokers, non-smokers, and people who vape? So that is the end of what I wanted to talk about now. I hope I've left us enough time to talk a little bit about things because I think that's a valuable part of these meetings.
Hey, let me start
Sure.
discussion. I have to leave in about 10 minutes. I think I heard you a while ago saying that Binghamton got the better Schertzer, but I think I
Well,
disagree.
I don't know about the better shirts. The older shirts they definitely got. That is unquestionable.
Now I
It's kind
think
of you
we
to
got
say,
the better
Satish.
one. We got the better
That's
one.
kind of you.
So I'm really impressed to see the kind of work you are doing. There's a lot of fundamentals, but at the same time, there is a good mix of applications and so on. And I know all this is possible only because you have help at home.
I do have
And
lots of help at home, and I'm eternally
yeah, it's
grateful.
super hydrophilic, super
Yeah, right.
hydrophobic,
So
stop
just
stress.
to fill everybody in, so I had a chance to go to Norway to this conference that Satish was running, and if I didn't bring my family, the locks would have been changed at home. So everybody was there, and Gabrielle, our eldest, was teaching everybody about super hydrophilic and super hydrophobic surfaces when she was there. So it was pretty
Yeah,
fun.
that was pretty cool. Pretty cool. So, I mean, what I think we could do is, by the way, since Michael wasn't coming, we canceled the Norway conference this year. Uh,
I
Michael,
think that's
so
the only reason,
maybe,
right, Satish?
yeah, maybe, I'm thinking we are having so many cool things going. How about we going in a large group? And I'm the organizer, so you're all invited. And we can really make a good showing. And then there will be many people from outside who would be coming. And then you can form these collaborations. And there are people from all over the world and obviously from the top universities here. And I think that would be an excellent opportunity. Looking at some of these things, there are many, many things that I'm personally excited about. But I'd like to, see, and I will talk to you when there are some things where I can contribute, but very good work.
Thanks, Satish.
So, with that, I'll let you guys go. I'll just listen in until my caller calls me.
I appreciate your
So,
feedback, Satish. Thank you.
yeah, and
Yeah. Hey, nice work, Mike.
Thanks, Stephen.
This is like being in a candy store here. I mean, you got a lot of good pickings here. So, what do you think you want to do? At some point, you got to put you got to dig in deep, right, into some of these
Yeah, but I
things.
think so. I've had this feedback before too, Stephen. My opinion, and maybe it's true, maybe it's not. My opinion is that we're digging in deep on the fundamentals, right? And then that allows us to then help contribute with people who are experts, right? So like I know about droplet manipulation, but I don't know about CRISPR detection, right? Thankfully, there's somebody whose office right down the hall knows about CRISPR detection, right? So I think what I try to do, and I'm not saying that I'm perfect here, but what I try to do is understand the physics of what I do, right? And then there's some ways where that contributes sort of to the core of what I do. But then, if I can work with Patricia on lubrication stuff, certainly I wouldn't be leading that project, but if I can chip in, then I think that's valuable.
No, it's just an issue of hours in the day. I mean,
yeah, that's definitely. Well, we got lots of time now, right?
You gotta sleep,
I'm early to bed, right?
But nice presentation.
Thank you very much.
Nice work.
since no one is asking any questions, and the call hasn't come in yet, let me ask you a question here.
Mm-hmm.
How can you, how much contact angle change you can accomplish given any choice of material and
this is a good question, Satish.
liquid?
So I think that this is why we put the hydrophobic surface down. So it depends on, basically the higher your initial contact angle is, the more change in contact angle you can get. So
can I get almost super hydrophilic?
So what I've seen, so from Teflon, which is about 120, I can get it down to about 90. Eventually you get to contact angle saturation, where, so if your contact angle keeps coming down and down, eventually that vector is not pulling out enough. Nobody really understands the mechanism for why it saturates, but it follows the curve and then...
Starting with hydrophilic.
Yes.
Can you make it super hydrophilic?
I think that you would have a tough time changing it, because I think if it already starts as hydrophilic, to make it more hydrophilic is difficult.
Yeah.
This is not exactly the question that you're asking, but there was a recent paper that was talking about electro-de-wetting, where you talk about a droplet on a hydrophilic surface. Now, it doesn't have any of the polymers on it. You apply a voltage, and if there's sort of a polar surfactant inside the droplet, you can make a hydrophilic droplet look more hydrophobic. So I know that's different than what you're asking. I don't think that if you start with a very hydrophilic droplet that you can move it much more in the hydrophilic direction.
Okay, so going hydrophobic is an easier route.
So it's easier to make something hydrophilic, and then it looks like there's some very recent work where you can take something hydrophilic and make it a little bit more hydrophobic. So electro-wetting or electro-de-wetting, but it's hard to go further if 90 degrees is here and 120 is here, it's hard to go. It's easier to go towards the middle. It's harder to go towards the sort of edges.
What are the outstanding fundamental questions that the community in your field is grappling?
Yeah, it's interesting, right? Because there's a lot of application-specific things, right? So, how can we do things like optimize electrode sort of signaling, right? Because it's nice because as we scale devices up, then you don't have this problem of having to make more and more channels. But eventually you have to have more and more signals, right? So that's one of the reasons why people do these three-dimensional devices and they're moving them up and around. So I think in our area, then there's also a lot of biological questions, right? So, which kind of materials can you be moving around, right? How can you be automating things? I think this droplet jumping thing is a relatively new thing. So people are trying to understand the dynamics of the droplets as they move around. And we're only applying a pulse. So you can apply different waveforms and maybe get standing waves on the droplets and get some sort of interesting patterns. How do you get more? So do you know what a TFT is? So this is like on your cell phone, you have sort of this electrode array that's very, very small different electrodes, right? So you can like drag your finger across and it measures, right? How do we sort of bring that kind of a sort of fabrication technology into the space? But that's really hard to do making things at a university, right? So I've talked to, right? So there's Professor Hirschman here has some contacts at Corning that make TFT devices. So that's something that we're interested in sort of looking at as well.
Okay, great. All right, I'll connect with you and others next week. So I'm signing.
All right, thanks, Satish.
Thank you.
So, I think that I know about droplets, but I guess I know a little bit about a bunch of other stuff too. So if you're interested in, I taught a little bit in mechatronics when I was at U of T, so I know a little bit about that. But again, it's not my sort of area of expertise.
Hey, I had one question. Yeah. This came back to the maybe your tenure presentation at some point.
Yeah.
That saturation curve there.
Yeah, this is a good question.
We had this discussion, I think, a few years ago. It just hit me that we discussed it. I don't know which slide it was on.
I don't think I have a slide of it, but I'll draw it for you. 1 second.
There's a curve that flattens out, and the data goes all over the place.
Yeah. So
you had it up there.
Right. So if I draw, excuse my drawing here. Right. This is a problem with these e-tablets. Right. So this is the apparent contact angle when you apply a voltage.
Yeah.
Right. And this is the voltage that you apply down here. What the theory says is you get this kind of a curve that looks like this. What the data says is this works up until some point, right?
Yeah.
So this comes down like this, and then you can't sort of infinitely reduce the contact angle.
Yeah.
Right?
And then the data was all over the place, I remember, in the flat
Yeah, it kind of
area.
mostly gets flat here. Maybe it still goes down a little bit. There's a lot of different explanations for why this happens. So some people have seen, in some cases, you get a droplet and now you're pulling on it so much that you actually are ejecting small droplets from the surface. There's some other questions about it's just so the vector is always kind of going perpendicular to the surface here, right? So as it gets flatter, you're just losing force that goes further. You know, so this is kind of an open fundamental question, but it definitely happens.
Yeah. So no one's got, since the last time you presented that, no one's really answered.
I have not seen a definitive answer. I'm sure that there could be many different mechanisms depending on what the fluid type is or something.
Yeah, that's interesting. I just was wondering. It's one thing I remembered back from when you presented before, this saturation.
Mm-hmm. And actually maybe I should have said this to Satish too when he asked this question. This maybe right now doesn't feel too much like if we really knew this answer, we could make so I think that the community kind of feels like it'd be nice to know, but what's the benefit of knowing? Yeah. But sometimes you don't know what the benefit is till you know the answer, right?
Yeah, it's just purely academic interest here. You got something that can't be described.
Yeah, it's certainly
your current theory.
It's certainly interesting, and I mean the theory works really well until it doesn't, right?
Yeah. Now you know where it doesn't. It's interesting.
Yeah.
Mike, I have one question. So when I work on soft material cutting, okay, so in the very beginning, we need to break the surface.
Yep.
Okay, to use blade to just like break the surface. So we really care about surface energy, or how much energy we need to break the surface. So I'm just wondering, what is the best way to narrow the surface, energy? We try to use a contact angle to estimate surface energy, but used to be much lower than you know the actual surface energy.
Yeah, so the way that
have
I would normally measure surface energy is like that, with a pendant drop, or maybe like a droplet on a surface, and you can do a series of different measurements on known surfaces and then on whatever surface that you have. I mean, I can go over that with you if you'd like. We can talk about that a little bit.
Okay.
I don't know if it would. So you're trying to measure the surface energy of this. It's almost like a gelatinous material, right? Like something like PDMS that's an elastomer.
Right. That's correct.
Yeah. So I mean, we can sit down and sort of think about that. Yeah.
Yeah. So we we always need this surface energy to use to analyze our cutting process. Unfortunately, the energy estimated by the contact angle always be much smaller. I just like to not know what's the reason. Probably that is not the very accurate way. Yeah.
Well, it's tricky with
Probably
contact angles because you know, especially if you're using PDMS. Like PDMS has a reasonably high contact angle hysteresis, so it's really hard to measure the equilibrium contact angle, right? So if you just put a droplet on, so here, I think I have a slide that kind of shows this a little bit.
You can maybe do the experiment. I got some Teflon downstairs
Here,
somewhere.
right? Yeah, right. You just I wouldn't use that pan. Right. So
I
here,
got some Teflon.
if we have a droplet and we add fluid, the droplet's going to get bigger at first, and the contact line's going to stay pinned, but eventually it's going to start moving, and then there'll be some contact angle that stays pretty constant as the droplet is advancing. We call that the advancing contact angle. Now we can have a droplet and we can suck fluid out, and the same thing, the friction on the surface is going to keep the contact line there until it eventually moves in, and we'll measure a receding contact angle, right? So there's this band of contact angles between the receding contact angle and the advancing contact angle, and the equilibrium contact angle is in between that band. So, so if you're just putting a droplet down on a surface, you can, right? You might get an equilibrium angle that you're measuring, or an initial angle that you're measuring, but you're not sure if that's at equilibrium, especially if you have a surface where there's a wide difference between the advancing and receding angles. For the experiments we've done on PDMS, there's a pretty reasonable gap between the receding angle and the advancing angle. So, but I can talk to you a little bit about ways to sort of estimate the Young's angle or the equilibrium angle from the advancing and the receding angle, if you'd like to talk about that. I mean, we can try. So we had to do the same thing here, right? Because if you look, this line, we need to know where this intercept is, and this intercept would be our estimation of that equilibrium angle, right there. So there's ways that you can kind of estimate that.
Okay. Yeah, definitely. I will follow up with you.
Yeah, that sounds good.
Yeah.
Thanks.
So nice job, Mike.
Thank you, guys. Appreciate it.
Okay, I'm gonna get going.
All right.
So have a good week.
Yeah, you too.
Thank you,
Take
Mike.
care. Thank you.